Ever since Fleming noticed a strange clearing zone on his petri dishes, humankind has used antibiotics based on fungi to fight and destroy the diseases that once tyrannized our ancestors. In fact, antibiotics were so effective at treating humans that we also decided to use them on our livestock, to such an advanced degree that agriculture now accounts for 80% of antibiotics used in recent years. Hello, my name is Alex Kelly and I grew up on an Angus beef grazing property in regional New South Wales. As I grew up, my father would constantly fret about the impossible catch-22 he was trapped within. A vancomycin treatment would improve the growth rate of our herds, but even in the bush, we understood the pressing concern of antibiotic resistance. Such widespread and indiscriminate use of a broad-spectrum treatment has had the inevitable consequence of building immunity within bacterial population. Today, diseases that are resistant to our most powerful treatments are a regular feature in hospitals. It is merely a lucky coincidence that a truly virulent and deadly disease has not yet inherited this resistance. Even if we reach 2050 without a devastating crossover event, the global economy will lose $2.1 trillion from attempting to treat human patients affected by superbugs. The agricultural sector faces a far more pressing situation. If an antibiotic-resistant strain of bacteria starts to spread through a national herd, the requisite culling will leave most of the industry in an irrecoverable state.
While this pressing problem occurs at the global scale, Agriphage will attempt to provide solutions to the Australian grazing and poultry markets with the option to scale towards global distribution when the business model succeeds. Our target market will be the Australian livestock industry, and our proof of concept will be the bacteriophage cocktail targeting bovine respiratory disease in beef cattle and infectious cryozoa in chickens. These two common diseases are rarely lethal, but significantly decrease the growth rate of effective livestock. It is common practice around the globe to supplement the diets of livestock with antibiotics to suppress these infections and improve the growth rates. However, the Australian government restricts the industry from using the most powerful antibiotics because they rightly fear the consequences of resistance spreading to human diseases.
Agriphage plans to develop a protocol for rapid screening and testing of native bacteriophage populations that have already evolved in the grazing ecosystem. For example, taking water samples from a trough that BRD-infected cattle have been drinking from should yield 10 to 100 different species of phage that attack the BRD bacteria. If we can isolate and purify just 5 of these into a bacteriophage cocktail, we will have a highly effective treatment for the disease with almost a 0% chance of immunity being developed. This makes the competitive advantage of a bacteriophage therapy over antibiotics immediately clear.
This cornucopia of competing pathogens will each have a unique approach to attacking and killing the BRD bacteria, which means that a cocktail of 5 phages will require 5 simultaneous mutations from the bacteria if it wishes to survive. The specific nature of this infectious virus is such that it is incapable of killing the native microflora that is wiped out by the broader spectrum antibiotics, which would theoretically improve growth rates even more than the blanket antibiotic treatments used by most feedlots. This specificity also ensures that there is no evolved capacity for the virus to affect the mammalian cells of the cattle or of the grazer. Finally, the quantity of novel antibiotics being developed has slowed to a trickle. A system such as this that locates newly evolved viruses in nature has exponentially more potential for the development of multiple effective therapeutics.
So how are we going to do it? I'm sitting here in the BioFoundry, a not-for-profit biotechnology lab that does bacterial and mammalian cell work. We plan to use E. coli to propagate our phages in order to make a clinical dose, so this research project is simply not compatible with this space. The Agriphage team will need to build a new laboratory workspace to develop this protocol, but fortunately, the BioFoundry already owns most of the equipment that would be required to fill out such a space. Fortunately, the lack of genetic modification in this developmental model will preclude the need for OGTR approval. Water samples will be obtained from the water troughs at Rugby Station during a BRD outbreak and from Cordina chicken farms during a coryza outbreak. As these occur during multiple points of the year, we won't need to wait long for an opportunity.
Next, we'll build a library of bacteriophages, attempting to characterize the major players in the viral ecosystem. Using RT-PCR, we'll identify the phages that affect our target bacterium from plaques grown in E. coli in the lab. A serial dilution of the plaques will eventually yield a pure viral sample that will act as one of the seeds for our therapeutic cocktails. These two diseases are just the start. Russia and Georgia have consistently cured human illnesses using bacteriophages for over 70 years, and there are no limits to the potential bacterial diseases we could target in agriculture. Whilst we have chosen to cure animals instead of people, the vital importance of this research to preventing a global human pandemic must not be forgotten.